Hello everyone, in this video we are going to discuss about Streptococcus viridans. So Streptococcus viridans, it is a gram-positive cocci which is arranged in chains. As you can see in this picture, it is also alpha-hemolytic like Streptococcus pneumoniae. So we know the Streptococcus is divided based on the hemolysis on the blood agar into alpha, beta and gamma groups. Alpha hemolysis means partial hemolysis and beta hemolysis means complete hemolysis and gamma means no hemolysis. So the Streptococcus viridans, it belongs to the alpha hemolysis group along with the Streptococcus pneumoniae, and it is also optochin resistant. In contrast to the Streptococcus pneumoniae, which is also a alpha hemolytic group, how we can differentiate it from the viridans is it is optochin sensitive. It is also bile insoluble, unlikely to the Streptococcus pneumoniae, which is bile soluble. And this viridans is present in the normal oral flora, like we can see in dental plaques and also in gingiva. So coming to the various types of the viridans. So this viridans is a group of organisms. So the species which are present in the Streptococcus viridans group are Streptococcus mitis, Streptococcus salivarius, sanguinus, mutans and anginosus. So these organisms causes different things. Mitis causes bacteremia, whereas mitis and salivarius causes subacute bacterial endocarditis, sanguinus causes deep tissue abscess, mutans causes dental caries and anginosus causes brain and liver abscess. So coming to the virulence factors of this viridans group, these produce dextrans. These dextrans will help to stick to the fibrin platelet aggregates. So these fibrin platelet aggregates, these help to stick to this and on the damaged valves. So these attacks mostly the prosthetic valves. Next, coming to the clinical diseases which are caused by this Streptococcus viridans are these causes dental caries, subacute bacterial endocarditis on damaged walls after dental work. So diagnosis is based on the gram staining, where we can see gram positive cocci arranged in chains, alpha hemolysis where we can see partial hemolysis, and optochin resistant. So whenever we place a disc of optochin on antibiotic sensitivity testing, whenever we place this disc, whenever the growth is complete, no stoppage of growth is there, then it is called as optochin resistant. So contrary to whenever there is a zone of inhibition of the growth around the disc, it is called as optochin sensitive. Sensitive one is pneumoniae and resistant one is viridans group. So coming to the bile insolubility, whenever we grow something on the bile like Streptococcus mitis, which is a part of a viridans group, it is not lysed by the bile, whereas the pneumonia, which is the part of alpha hemolytic group, it is lysed by the bile. So how we can differentiate this alpha hemolytic group on the versus the, that is pneumonia versus this viridans group, is the viridans is bile insoluble, optochin resistant. So these are the two differentiating factors. So coming to the treatment, penicillin is used for susceptible strains, and prophylaxis is must and should for the before dental procedures in high risk patients like in prosthetic valves. So we can easily remember this viridans based on the mnemonic of viridans only. V stands for viridans group, I stands for infects the damaged heart valves, R stands for resistance to optochin, I stands for insoluble in bile, D stands for dental procedures leading to infections, that is DAN, S stands for subacute bacterial endocarditis. So subacute bacterial endocarditis, it is most common. These are the most common things which are caused by this viridans group. So these are the important points in Streptococcus viridans.